Hello and welcome. I am your moderator for this joint presentation from Illumina and Genomenon, Genomic Biomarker Selection for Companion Diagnostic Development. My name is Mark Kiel, and I am the Chief Science Officer and founder of Genomenon, where we are organizing the world's genomic information to drive clinical diagnosis and drug discovery. We have a full agenda to cover today and a great lineup of panelists. As a reminder, this webinar is being recorded and will be emailed to you after the event. Also, you can submit questions in the QA box in the Go-To panel, and if we have time, we'll cover some of those audience questions after our panel discussion. The themes of today's discussion will be the role of genomics in companion diagnostics, the challenges attendant with this relatively new discipline, and emerging trends for genomics in companion diagnostics with a focus on oncology. We will begin with a brief introduction from Dr. David Eberhard, the Senior Medical Director for Oncology Diagnostics at Illumina and our co-sponsor. David, I invite you to turn your camera on and to begin. Just let me know when to advance the slides.
Thank you, Mark. It's my pleasure to be here today. And today our conversation is going to be about precision medicine. And I really like the definition provided here. The use of therapeutics that are expected to confer benefit to a subset of patients whose cancer displays molecular or cellular features, specific features. And these specific molecular or cellular features are what we're talking about when we talk about biomarkers in cancer. Next slide, please. This shows the rapid pace of the development and approval of biomarker-driven cancer indications over the past 2 decades. And really the first biomarker kind of driven cancer indications were even before the turn of the millennium in breast cancer, where ER/PR status was used to select anti-hormonal therapy for breast cancer patients. And also in breast cancer, the development of the anti-HER2 antibody Herceptin, which has marked efficacy in breast cancer patients whose tumors overexpress HER2 or have amplification of the HER2 gene. This slide we're looking at since the year 2000, the explosion that really was catalyzed by discoveries in the lung cancer field. And particularly lung cancer has been revolutionized by genomic biomarker discoveries over the past 2 decades. EGFR inhibitors, gefitinib and erlotinib, were introduced for the treatment of lung cancer early in the 2000s. And just about the time that they were being introduced, discoveries were made about EGFR mutations which sensitized lung cancers to treatment with these inhibitors. And EGFR mutant lung cancers displayed remarkable sensitivity to these inhibitors. Subsequently, there was a huge amount of attention given to identifying the genomic landscape, describing the genomic alterations in lung cancers and other tumor types. And these genomic landscape discovery efforts resulted in the identification of several key driver oncogenes, which are mutated with some frequency in lung cancer. And these are shown to the left, including RET, MET, ALK, EGFR, BRAF, NTRK, etc. Subsequently, over the next 2 decades, after these driver gene mutations were discovered, selective inhibitors against these driver genes were developed and brought through the approval process and are now coming onto market as targeted therapies which are approved for use in lung cancer patients who specifically have these particular mutations in these driver genes. And we can really see the fruits of these discovery and development efforts coming about in the right-hand side of this figure in the past decade, the approval of more and more biomarker-driven drugs and companion diagnostics for patient selection. Likewise, the development and introduction of immune checkpoint inhibitors, which again are targeted therapies against particular molecules that modulate the immune tumor cell interaction. And the use of these therapies are also driven by the use of biomarkers to assess the presence of these targets in tumors. Next slide, please. And lung cancer has really shown us what can happen when we understand the molecular landscape and the mutational landscape and develop drugs that target these selective mutations in tumors. And we're seeing this beginning to happen in other tumor types as well. And here's a figure showing a variety of other tumors for which particular genes are mutated and drugs are being developed with, again, remarkable efficacy when patients are selected appropriately with biomarker tests. Some of these biomarkers are specific for particular indications. Others are found in one or two or more different tumor types. And most recently have been the approvals of drugs in solid tumor indications which are defined solely on the basis of a particular biomarker, not on the tumor type itself. So these drugs include, for example, pembrolizumab, which is approved in solid tumors which have a high tumor mutation burden or high MSI status, or NTRK inhibitors for solid tumors which have NTRK fusions. And again, in these cases, the tumor type doesn't matter. The key thing is the presence of the targeted mutation. Next slide, please. So oncologists and patients are really enjoying the fruits of these drugs, which are targeted against these particular mutations that drive their tumors. The biomarker testing is done by the pathology laboratory, and the incredible pace of development and adoption of these different targeted therapies has created a challenge for the pathology laboratories, which have been how to best do testing for the biomarkers which are associated with use of these targeted therapies. And these biomarker tests are necessary in order to appropriately treat patients who have these alterations. The traditional approach of the solid tumor pathologist has been to cut sections of the tumor onto slides and perform a test on each slide. And traditionally, this has really been a single gene testing approach. And this could be an IHC assay, a fluorescence in situ hybridization assay, or a single gene molecular mutation assay. With the adoption of more and more drugs that require testing for more and more different genomic biomarkers, this has created a bit of a challenge in how to best perform testing for the variety of biomarkers which are possible. And if we were to use the traditional approach here, this might be a single gene approach where each desired biomarker is tested. The result is returned to the pathologist. And if that result is positive, great, the recommendation can be made for patient treatment. If the result is negative, then we go on and test for the next biomarker. And if that's negative, we go on and test for the next biomarker. And so this is a serial, iterative, reflex kind of an approach. This approach has some disadvantages when we have such a number of potential biomarkers that need to be tested for, and particularly if the biomarkers are present at a relatively low frequency in the population. And that is that tumor samples often are small and limited, and by the time we get through each of these tests, we may run out of sample before we've been able to do all of the testing that is appropriate for that particular patient. Secondly, it takes a lot of time to go through this iterative process, performing one test, waiting for the result, performing the next test, waiting for the result. And so if we went through the entire panel of genes, for example, that are approved in non-small cell lung cancer, this could take up to 6 weeks to perform testing and up to 29 slides if we have that much tissue left from that lung cancer sample. Next slide, please. So Jeff Conroy from OmniSeq and Roswell Park summed it up as in non-small cell lung cancer, The NCCN guidelines recommend a multitude of biomarkers be tested, and in some situations, this could require at least 30 slides. And particularly in lung cancer, the available tissue may be only a very small biopsy, and we frequently run into situations where we run out of tissue before all of the NCCN guideline recommended testing has been completed. Next slide, please. Molecular pathologists have been working on how to address this challenge of serial testing, which results in tissue depletion and can take an unacceptably long time in order to perform all the required testing. And what I'm showing here are 3 boxes. In each box, there are different gene variants, small variants, CNVs, fusions, splice variants on the left, and then a variety of genes which would need to be tested in non-small cell lung cancer. And the top box kind of shows the single gene test approach that I was just talking about, where, for example, in this patient, if we wanted to test for a ROS1 fusion, we would perform, for example, a FISH assay. And that one assay returns that one single result. From the grid, you can see that there are a lot more results that might be appropriate and important for the treatment of this patient. And so we want to be able to better assess all of those. So next-gen sequencing, or NGS, has enabled the development of gene panels. And the first panels that were developed are small hotspot gene panels where portions of the genes of interest are sequenced, the hotspots where mutations that are of clinical interest most likely occur. And this allows us to assess a wider number of genes of interest and gene variants of interest at the same time, but it still can miss those, particularly those less common gene variants that ultimately may be important for informing the management of any particular patient. And ultimately, where we're going and where we are today at the cutting edge of molecular genomics for therapeutic selection in lung cancer and other cancer types is comprehensive genomic profiling. And this is large-scale next-generation sequencing-based testing, which enables the assessment of all of the genes of interest and all of the different variant types in those genes that might be relevant at the same time, so that the testing result for all variants, all genes, is returned at one time from a single test from a single tumor sample. And so this can result in providing the most information, the most efficiently using one tissue sample, performing one test, one and done. Next slide, please. A quote from Jerry Valentine from Intermountain Health. The change from a single biomarker testing to comprehensive panel approach has been driven by a combination of factors that include inherent efficiency of a single comprehensive panel, which is key among cancers and other sample types that have limited tissue. Next slide, please. And evidence from real-world experiences starting to accumulate and be published that shows that this concept of genomically matching patients to targeted therapies or immunotherapies does result in patient benefit in real-world settings. And here are a few studies looking at lung cancer. On the left is a study from 15 community oncology centers comparing molecularly matched therapies with cytotoxic chemotherapies and demonstrating that matched therapies, and this is a biomarker matched therapy, led to higher overall survival, 31.8 months in the matched therapy group compared to 12.7 months in chemotherapy group. On the center, another study of over 1,000 non-small cell lung cancer patients, again comparing molecularly matched targeted therapy regimens to non-matched regimens. And again, looking at overall survival, showing that matched therapy patients enjoyed at 18.6 months overall survival compared to the non-matched therapy arm, which was 11.4 months. And on the right is an interesting study. It was a retrospective study looking at 101 lung adenocarcinoma patients who received comprehensive genomic profile testing And of these patients who received comprehensive genomic profiling, 50% of those patients actually had a match therapy chosen as a result of that testing. And among those patients who received matched therapies, the overall response rate was 65%. So this really shows that routine testing using comprehensive genomic profiling can quite often, 50% of the time, deliver a result which is actionable to the oncologist and the patient, and that results in a terrific response rate. Next slide, please. So because of this evidence that comprehensive genomic profiling, next generation sequencing testing does benefit patients in oncology, large panel NGS tests are increasingly being recommended by the bodies in the US, such as NCCN, and in Europe, such as ESMO, for a standard of care assessment and treatment of patients. So for example, in NCCN solid tissue guidelines, the wording is a little bit different in different guidelines. NGS testing is recommended in quite a number of different solid tumor indications now, shown on the left. More specifically, comprehensive genomic profiling or broad molecular profiling panels are recommended in 6 different indications at this point in time, and these are increasing fairly quickly. Also, tumor mutational burden testing is recommended in the center in quite a number of different tumor types as well. TMB requires a large panel NGS test in order to be performed appropriately. 1 megabase or more of genomic sequence has to be interrogated in order to appropriately assess TMB. So for these TMB recommendations, the implication is that NGS CGP needs to be performed. And likewise, the ESMO recommendations are going the same direction for targeted panels and CGP broad molecular panels. Next slide, please. Along with solid tumor testing using next-gen sequencing, comprehensive genomic profiling panels, really exciting development is our ability to use these same types of broad molecular assays to interrogate blood samples from solid tumor patients, where circulating tumor DNA that's released from solid tumors into the bloodstream, can be isolated and sequenced, and the tumor-specific biomarkers that can guide therapy can also be identified in blood samples as well as tissue samples. And our ability to perform this kind of testing in liquid biopsies, blood samples, is complementary to our ability to test tissues. Right now, we're viewing this in pathology as these are not replacements one for the other, but they really are complementary techniques. An example, if we run out of tissue, earlier we were talking about how tissue samples can be limited in quantity and may not have sufficient tumor tissue or tumor cells available for testing. This is a situation when liquid biopsy can be very useful. Or if a patient is very frail, medically unfit for an invasive biopsy procedure, a blood sample could be used instead. And finally, liquid biopsy has the potential for drug developers to have a more simple sample collection procedure where patients can be screened for eligibility for enrollment into their molecularly guided clinical trials. Next slide, please. So to summarize this introduction, comprehensive genomic profiling enables precision medicine by allowing us to comprehensively assess clinically relevant biomarkers that guide the use of targeted therapies and immunotherapies. And CGP, comprehensive genomic profiling, can assess pan-cancer indications, a variety of different tumor types, a variety of different molecular targets, DNA, RNA, the different variant types that we talked about earlier. CGP allows the consolidation of all of the different possible tests into one test that covers everything. Sample types can be either tumor tissue or liquid biopsies that look at circulating tumor DNA in the blood. These results maximize our ability to identify actionable variants, and clinical utility is now being shown that CGP leads to molecular matched therapy selection and clinical trials with better clinical outcomes for cancer patients. So thank you for allowing me to introduce this.
Oh, that was great, David. I invite you to stay on with your camera and join the other panelists as I introduce them. Other panelists, as I call out your name, please turn on your video and unmute your microphone, and we'll begin the conversation. So I will introduce you by title, and I'll ask you to introduce yourselves by way of background. Abdel, welcome. Abdel Halim is the Vice President of Biomarkers and Companion Diagnostics at Taiho Oncology. Scott Patterson is the Vice President of Biomarker Science at Gilead. Scott, if you'd join us. And I think it's not too familiar to call you Nino. Nino Cerecci is the Vice President with Diagnostic Development and Med Affairs at Loxo Oncology. So welcome, everybody. Thank you, David, for that great introduction. I have about 15 extra questions that I've got besides the ones that I had come prepared for. But let's begin by having the panelists introduce themselves. I'll start with you, Abdel, if you wouldn't mind telling me what you do at Taiho and what your experience has been.
Sure. At Taiho, I manage the biomarkers and companion diagnostics, and currently I'm running 2 companion diagnostics for registration, including actually both of them are next generation sequencing, and for panels 500 plus genes. And also, in addition to that, I am enabling patient selection in early phase clinical trials, in many clinical trials, including next generation sequencing. And in addition to patient selection, as Dr. Eberhard just said, genomic profiling is now becoming more important for clinicians to manage patient above and beyond targeted therapy, because enable the clinician to know the prognosis or to have an idea about the prognosis and patient monitoring using local biopsy. So I lead all these aspects at Taiho. Thank you.
Great. Scott.
Great. Thank you, Mark, and great to be here. Thank you for the opportunity. So I have the privilege to lead a department called Biomarker Sciences at Gilead that's responsible for biomarker translational work and diagnostics across all therapeutic areas, from research to all phases of development, as well as into post-marketing. We also have currently a registrational study with patient selection using NGS. My background is I had 13 years in academia, finishing up as a faculty member at Cold Spring Harbor Lab. I joined industry 29 years ago into discovery research, the last 18 being involved in biomarker and translational work, and the last 14 to 15 where that's spilled into diagnostics, obviously from the pharma side. And I've been lucky enough to be involved in a couple of PMAs, de novo 510K, and multiple CE-IVD partnerships for a range of analytes, including therapeutic drug monitoring, soluble proteins, tissue-based, and genomics, including, of course, NGS. And it's a privilege to be on the panel. Thanks for the opportunity.
Great. Welcome, Nino.
Hi, everyone. My name is Nino Cerecci. I lead biomarker development and medical affairs at Loxo Oncology at Lilly. My group actually spans the gamut from early phase development, focusing on biomarkers and translational work, through patient selection and CDX selection, through post-marketing and medical affairs as it applies to the diagnostic in the clinic. We have currently one CDX partnership, one asset that has multiple CDX partnerships, including one with Illumina on the TSF-500 and others in development in the pipeline. Prior to joining Loxo, I was a molecular pathologist at Columbia University, did clinical testing, worked with the Illumina platforms and thermal platforms in my laboratory. So have a very practical sense for what it means to actually bring on these platforms internally, to struggle not only with the technical aspects of the technology, but also to struggle with the financial burden of bringing on these platforms, particularly in the setting of limited reimbursement for non-CDX platforms. So happy to be here.
Yeah, great. And Nino, I'll start with you with the first question. So we want to get to emerging trends toward the end of our discussion, but let's begin in the past. And Dr. Eberhard set it up quite nicely. Has NGS made your jobs easier or harder? And what are the ramifications of NGS overall from a patient improvement of treatment perspective? So Nino, you alluded to some of them. Net, has it been better or worse, or if a mixture, clarify?
Yeah, so I'll put on my pharma hat first and say that, you know, there's an opportunity to actually utilize the CDX regulations and sort of the purpose of CDX regulation, which is to ensure quality for our patients and to help sponsors feel confident that they're selecting the right patients for their trials. There's an opportunity with the advent of large NGS panels. It's important to say that NGS panels that are comprehensive to actually be meaningfully useful within the clinical laboratory. I think in the past, CDXs that were single analyte or specific to one analyte, one drug, just were not reasonable to bring into the clinic. You know, it just took too much money. They're usually more expensive, too much time from a CLIA and uptake perspective. It just wasn't reasonable. And so no one used them. And I could say as a pathologist, I would never have brought on a single analyte CDX into my laboratory other than maybe BCR able. So the advent of kitted CGP assays like what Illumina produces, like what other manufacturers produce, I think actually helps make the CDX regulation and improve patient quality. From a clinical perspective, of course, I mean, this idea that you'd have to use minimal, or that you have to use bits of tissue for single analyte approaches in an era where we have 10 biomarkers in non-small cell lung cancer and growing in other cell types. From a clinical perspective, of course, it makes sense to sort of take the tissue and do the most with it that you can. The struggles or the pain points have really been around implementation, so that you know, getting high quality testing in laboratory, getting alignment on reporting structure and interpretation, and frankly, getting paid for the work has been a real struggle. And so even with the NCD by Medicare, you know, we see limitations from in coverage and reimbursement for large panels by private payers. We certainly see that globally across Europe and Asia, that reimbursement is still an issue for these larger panels. So we have a lot more work to do, but I think it is the right move from a quality and from a patient care perspective.
Great. Scott, let's focus a bit on the implementation aspects. I'm happy to let you build on any of the other points that Nino made, but if you could focus on what the challenges have been from an implementation perspective, and we can start to branch out the conversation from there.
Sure. Well, I mean, from the clinical trial perspective, obviously having a comprehensive panel is a significant advantage because you get to explore additional genes, which you may have some ideas around from an exploratory standpoint, but you wish to gain more data. So that's a clear advantage. The challenge that we face, of course, is one that, although we're thrilled to see that comprehensive genomic panels are being introduced into patient care as they should be, mostly as laboratory developed tests, creates enormous challenges then in the clinical trial space because, of course, centers wish to utilize their existing tests. That becomes a challenge then when you wish to use the test that's obviously the market-ready version of the IVD that you wish to file to register your drug, that will be the test that demonstrates clinical validity. And this creates challenges, and I understand those challenges, certainly from the clinical side as well. But it's something that really, until we're at the stage where there are authorized, registered diagnostics being used in all the clinical centers, we're going to continue to face this. I mean, that will be a great scenario when we have those platforms that are registered and we truly get to the stage of adding content, not adding content, but also adding clinical validity to biomarkers that are already part of panels that are being employed that are registered and authorized by health authorities globally. And of course, we're going to see lots of changes in Europe where it's going to actually raise that standard a lot. But that's where we see the challenges at the moment, but a really great opportunity for the future. And I think this ability to have the comprehensive genomic panels is one that many of us have wanted for a long time, and very happy that they're here. But some challenges to consider for implementation.
Yeah, when I was coming up in my research training, next generation sequencing was becoming a thing, and it was a great bonanza. And I was wearing a separate hat on the clinical side in my MD PhD training, and I recognized that there was a challenge to standardization and implementation and ensuring reproducibility across molecular diagnostics labs. Abdel, I don't know if you want to build on that implementation challenge, particularly from the perspective of standardizing the implementation of these assays as well as the interpretation.
Yeah, sure. I think I agree with all what has been said so far. Implementation probably is the current main limitation for next generation sequencing into clinical trials for companion diagnostics. No doubt that we have the science, we have the technology, and clinical value is proven. Clinician like to have as much as data about his patient or her patient, so that he or she can monitor patient, look at biopsy, all this type of things, prognostic biomarkers to at least to know if the patient has any Q mutations or Q occurring mutation. So this is very good to have, and very has been proven. Implementation, from my perspectives, has two folds. First one is how is the CRO, the clinical CRO, the CRO which enroll patients, is still working with the mentality of all comers. And when it comes to some of them, even including the major CROs, are still behind in understanding companion diagnostics and implementation companion diagnostics, approaching sites with ability to do companion diagnostics and targeted therapeutics. This is one aspect. The second aspect is the regulatory aspects. How to reach common ground between FDA and declare. FDA reporting and declare reporting and exploratory reporting. So because labs don't, especially the big organizations, I do like to name any, but you know them, still working also with the bureaucracy that they are not open for innovations, they are not open for adaptations, they are just like to report whatever the diagnostic companion diagnostic claim will be. For example, if it is EGFR T790M mutation, this will go in the claim. If the drug target this particular mutation, FDA say this will be your claim, and this will be your submission for IDE or significant risk determination and PMA. At the same time, the clinician like to have the data, the other data from the 500 plus genes. How to get them? There is two ways: either to report to them as under CLIA, which you need CLIA validation, which some clinical labs are reluctant even to go this path, or to report to them as exploratory biomarker, but to how to bridge the regulation to science. Science exploratory biomarkers, if you report to them under the umbrella of exploratory, this is not for medical decision. So who is going to provide this data to the clinician as the sites, and how the sites can use whatever named exploratory data for patients management? So implementation, as I said, to brief, is twofold. One related to mentality of the CRO and the big organization, CRO labs, laboratories. At the same time, how to reach a common ground between CLIA and FDA, and how to make the scientific data reach a clinician with no liability for legal liability or any aspects which you may have after that.
So let's build on the liability aspect. So David, one of the questions that I jotted down as you were talking about comprehensive genomic profiling was twofold. The challenge with a great richness of information, particularly if you're a pathologist. I was trained as a pathologist. It's different than on the genetic side, where the hope is that you can make a clear-cut interpretation from a yes or no analysis. And so can you speak to the challenge and how it's being addressed of dealing with too much information from CGP, as well as the risk that we might run from false calls? And then I'll invite everybody on the panel to weigh in as we start talking about the evidence at the variant level and how that feeds forward into guidelines and guidance for clinical interpretation. So David, if you wouldn't mind answering that first question.
Yeah, thanks, Mark. It's a great question, and it's very complex and rich. And so the part that I'll focus on relates to a couple of the panelists already talked about the challenges of validation. And I think it goes back to, what is validation? It's demonstrating the evidence to support a claim or an assertion that we wish to make. And particularly if that assertion is that there's a relationship between this biomarker, between this result, and the outcome of a treatment action based on that result, recommending a therapy. What should I do? What should I treat a patient with, given this biomarker result? And while scientifically that's very exciting and we can think about pathways and relationships and come up with ideas of what should be done or what we'd like to see tried, in the clinic, we really can't do it that way. We can't be so experimental.
We can't be cowboys in the clinic, I've said.
Exactly. And, this is, what I'll just right now say that FDA, I think, has been a great partner with us in the extremely complex challenges of developing, particularly comprehensive genomic profiling biomarkers, NGS. But there are a lot of, really complex challenges to overcome. One is the validation of results. Abdel talked about being able to return all of the results back to, the investigators in a trial in order to better understand, what's happening with those patients. Hugely important question, but, how do we validate those results so that we can trust those results? And then another aspect is once we have those results, what are the clinical associations that we're going to make? And particularly if we get to the point of making a recommendation, I'm going to do something or not do something. And there's a fine line between the returning of a laboratory result and the interpretation of that result. It's sort of the going into the art of medicine.
Yep.
And that is a line that FDA makes clear, that interpretation, annotation, et cetera, of results is something which really cannot be appropriately validated or cannot be validated using our current tools. And so we might separate those into, here's the result from an FDA approved test or an IVD device, and then with these results, someone needs to interpret those. And if we're developing IVDs, we'd love to have a product that we could put right in the oncologist's office, and they
Right.
would pump out a report, and that report would be crystal clear about what to do. But in fact, it's rarely that way. And we need experts, pathologists, geneticists, really an entire team who know about cancer genomics, who know about cancer patient management, who are familiar with each individual case, to come together and look at those results, discuss the results, and formulate recommendations for patient management. And this is a molecular tumor board. And I think, what we are really finding now that we have the ability to generate these large-scale genomic results, a big challenge is making these understandable and useful to the clinician, and not being cowboys, as you said, Mark, doing this in a very thoughtful way that does benefit patients. And just now we are, at Illumina, we're and other companies are developing software solutions to really try to make this happen most efficiently and to deliver end-to-end solutions. But I think right now I don't envision that there is available a software approach to replace the molecular tumor board. We can bring that data to the experts, but we need those experts to really synthesize the data and make the recommendations.
Well, Scott, I think you're about to jump in. Is David correct? Are we going to continue to deal with ambiguity in the interpretation, or will we reach a point where we as a field have collected enough evidence at the variant level to rule in for treatment or diagnosis specific patients based on their genotype? So that question or some other question that you had in your mind.
Sure. No, thanks, Mark. And yes, there'll certainly be cases where you still need that evaluation, and particularly when it's something that's outside of the clinical validity that's been established around a specific gene and set of variants. The challenges we get, of course, and you know, we're grateful to the work that we've done with you, Mark, around this, when we have a gene with a very large spectrum of mutations, where you're never going to get the clinical validity on all of those mutations. That pipeline calling is actually what we see as the biggest issue between LDTs and companion diagnostics. And this is something that others have found, and it's really a challenge, and certainly will be more so revealed when you have these genes with complex spectrum of mutations. That is that really a deleterious variant? How are you going to interpret that? But when you're coming at it from the companion diagnostic side, it's got to be defined. So there'll be, you know, even in that, there's going to be some areas of ambiguity. How is that going to be handled? But again, I would raise the concern around pipelines being different. And by the way, this has been shown to be an issue even with very simple sets of variants. And the pipeline calling is quite disparate. I think it's a very tall order to expect that pathology labs have that bioinformatics expertise. It's very challenging. And again, this is why that's a particular concern that we've seen and others have as well. Like I said, let alone in complex genes, but those that have even very limited numbers of defined variants.
Nino,
yeah, I'll jump in. You know, I mean, I hear your point, Scott, and understood for the majority of clinical laboratories that exist in the community. I think as a molecular pathologist, like, our training is in fact to assess variants that don't fit the general, you know, criteria of pathogenicity or the approved list of variants that exist on a label. And so I actually find CDXs valuable, again, if they're implementable within a laboratory and useful within a laboratory. But I do find that the lack of flexibility within that first page of a report can be actually quite damaging. And there's literature that suggests the opposite of what you described, which is to say you can be limited in what you find on a CDX if you're not looking for those other variants that are outside the scope of what's in the label, that for which maybe that variant hasn't been included in the clinical trial, but has every biological reason to be considered activating. And so that's where I think tools like the Genomenon database, I think bioinformatics pipelines that can be tweaked and adjusted and with appropriate oversight from a professional, I think are actually quite valuable. You know, just speaking from my experience in the NTRK space and now in the RET space for some of our drugs, you know, there is a wide spectrum of potential partner fusions. There's a wide spectrum of mutations that occur in RET, and how we, you know, we didn't enroll all of those patients in our clinical trial, but it's actually not a service to patients to limit use of the drug to a specific list that were tried in the trial. And I think we need to behave like physicians and like scientists in the clinic and make the right choices for our patients. So it's just I agree with your concern around the pipeline in LDTs, and actually think that sometimes they actually are because of the flexibility can actually give our patients more opportunities.
Yeah, let me just clarify that if I could. So,
sure.
Because I probably didn't state that very well. I agree entirely that there can be additional variants that need interpretation, and that should absolutely be the pathologist's purview. What I was saying is that it's known and it's been shown that labs that think they can call even the defined variants get them wrong. That's my issue, not the other side. It's where it's actually defined. And there's issues because it's the pipeline they're using. As far as the interpretation of other variants, I agree with you entirely. I think that that's critically important. And the readout, the report, I think should not be restricted to only what, it should be stated what's, clinically validated, could be on the label. It's got to include that other information. So that I'm absolutely on board. And sorry I didn't make that clear.
Yeah, and I'll just, and I appreciate the clarification. I would say we're living in a world right now where even from a legislative perspective, we're trying to balance this world of kitted, CDX-based, FDA-approved platforms versus LDTs that are maybe approved by another mechanism. And I 100% and wholeheartedly agree that there is no standard as of yet. Even CLIA really lacks the sort of teeth to get into some of that, the detail that's required for pipeline analysis and the analysis of the rigorousness of a platform. I think the answer is standardization of performance criteria and all of us aligning as a field and what's required for the analysis of a tumor sample, not necessarily always submitting to a CDX. And I think if we were to agree on those criteria, I think we'd actually solve for the problem that you just outlined, Scott. So thanks for bringing that up.
Open to the rest of the panelists, do you see a role for regulatory bodies in helping sort this out and guide where this trend might take us to the betterment of the patient community and providing the most appropriate care, or do you see them as an impediment because of their historical experience not being up to date? Abdel?
Yeah, actually, I would like to emphasize more on the points about bioinformatics and the tool.
Sure.
I think there are three major deficiencies in the pipelines or bioinformatic tools that we are using now. First is the lack of standardization or lack of harmony between different bioinformatics. And each team, of course, claim that they have the best, and no one knows which is the best. Recently, I ran exercise. I took the BAM files, not even the raw data, the raw sequence. I took the BAM files from one of the nation leader in liquid biopsy, and I gave it to another lab to run them through their bioinformatic tools. And believe it or not, rate of concordance was about 1% between the variant detected by the two organizations. And as I said, one of them was the nation lead in liquid biopsy, one of the nation leads. The second part is the variant of unknown significance. Science evolving every day, and variants of known significance driven by some applications with no clinical validity most of time, as Scott just said. So I think Genomenon can play a very important role, and the similar organization can play roles on how to identify, how to put definition of variant significance, and how to link the detected variant at DNA or RNA level with biological implication from mapping and the signaling and others. So the last point is, sorry, I need to stop, but biomarker definition is also very, very discrepant between different labs. And when you speak with FDA, they always say, make sure that the biomarker, if you are going to use multiple labs, make sure that the biomarker definition is the same, which is very unusual to have the biomarker definition. For example, Illumina test gene fusion or rearrangement on RNA, but Foundation and the others tested on DNA, which makes the biomarker definitions completely different. So who can lead all this? Can FDA lead it? Can NCI lead it? Who can lead this, or CAB or somebody else to do this standardization between pipelines, biomarker definition, variant of allelic frequencies? We need somebody to take the initiative.
Any takers in agreement or disagreement? One question that I had to build on Abdel's VUS commentary is we're learning more all the time, and more is being promulgated into the literature. And we talked before about where the evidence is straddling where there's sufficient evidence or insufficient evidence to make a definitive call on an actionable variant. What are we to do with newly emerging information, especially if the FDA and regulatory bodies suggest that you need to lock this down, the biomarkers for inclusion a priori? How are we going to address that gap as new information is brought to bear?
I think, Mark, one opportunity, and this is difficult within the current regulations though, is that we need to be able to capture that data into EMRs and track those patients. We need to build up this observational evidence. And I think there's real opportunities then if you have confidence in the variant calls that are made and that that's in the patient's medical record, that you can start to accumulate this data over time to see what's happening. Is that an opportunity to be able to go into an iterative mode to expand knowledge around which particular variants are impacting patient responses, resistance, or actually hopefully getting a response? So I think it's very important to see how we can capture that information. And to the point raised before, it's critical that if we have harmonization, then we can be very confident in this data that's being accumulated, and greater confidence, therefore, in submitting that to actually expand what the claims are, and whether it's really worrying about the claims or more importantly about guidelines and patient treatment. But if we're able to capture that into EMR data, which it can then be retrieved following those patients, I think there's a great opportunity. But it gets back again to that previous conversation about how confident are we in accumulating that data, and being involved in other efforts where it's looking at samples to how can one actually try to test platforms based, irrespective of whether LDTs or companion diagnostic, test them based upon common samples. How's that most appropriately done? And then that point around the bioinformatics. But I think capturing that data into the EMR, to me, really could start to address the concerns that Abdul raised.
Yeah, I'll jump in and just say that what resonated really about what you said, Scott, there was the sense that there's one I guess issue, which is expanding the label and including those variants in the label, and there's quite another, which is actual clinical practice, and the one I'm most concerned about is actual clinical practice. And so how do we help our laboratories doing testing, even if they're using a CDX? How do we help them keep up with the literature? How do we help develop information and knowledge around novel variants? And that's a bigger concern. And I think your idea around tapping into the EMR is sort of resonates with me, because this is a problem in clinical trial world in general, right? It's not just in precision medicine. It's how do we learn more about our drugs during the post-marketing phase of development, and how do we then feed that back into our development strategy and feed that back to our clinicians. And I think that we have a lot to learn around how to capture and utilize real-world evidence and real-world data. That's going to be sloppy. It's going to be messy, but actually has a value to patients and frankly to our labels.
One final question, David. I don't know if you have your prior engagement that you have to drop off for, but if you're here, you can answer. Otherwise, I'll solicit opinions from the other panelists. David talked about this concept, it's not new really anymore, of treating the mutation or the biomarker independent of the tissue type. How do we see that complicating implementation and interpretation of the success of a trial, especially when you might have disparate results across different patient types?
Yeah, I mean, one thought is that it's an attractive experiment that has to be approached carefully. And I think that this, cancer genomics in the clinic has been one of these areas where we found that the more we know, the more we need to know. And we're very excited by the example of finding a mutation in a particular tumor type that can be treated effectively with a targeted therapy, like an EGFR mutation in lung cancer. But if you find an alteration in EGFR in another tumor type, will they respond the same way? And certainly there's been a lot of clinical research and clinical studies that have investigated this. Sometimes we find that it works great, and we can have, for example, NTRK fusions in a variety of different tumor types that appear to respond to NTRK inhibitors. And so that has developed into an approved pan-cancer indication. But there are other times when we've learned, oftentimes from these clinical experiences, that different tumor types have different underlying biologies which we don't fully understand. And so, for example, if we look at the RAF MAP kinase signaling pathway in lung cancer versus colon cancer, those seem to be different diseases in a lot of ways. And so, particularly when we think about, refractory to treatment or the emergence of secondary resistance pathways and mutations, that, colon cancers do things differently than do lung cancers when they're treated with these targeted therapies, particularly in, the RAF MAP kinase axis. And that's, one example. So we have to be careful about overgeneralizing too quickly. These are ideas which deserve to be treated, in a clinical research environment, and explored, because that's how we make advances. But we have to be careful not to get too far ahead of ourselves in assuming that what we think is going to happen clinically actually will happen clinically.
So I'm looking at the time. I'll invite other panelists to weigh in on that, or otherwise, since that is a forward-looking statement that David provided in his answer, give me some insight from your perspective about where this trend will take us and what kind of complexities might we encounter as we mature the use of genomics and companion diagnostics. I'll begin with Nino.
Sure. So first of all, thanks for inviting me to the panel. It's been a really good discussion. And David, always a pleasure to hear about what's going on in Illumina. From my perspective, I think the direction CDX regulatory oversight of assays is going, and as it applies to trial enrollment, let's start there, is actually to probably move away from a single test, single approval to class-based approaches, class-based tests and approval, and frankly, setting criteria for performance rather than approval of specific assays. It's more reasonable. It actually meets sites where they are. It increases patient access to testing, high quality testing, and allows trial sponsors like my company to actually accrue patients at where they are, rather than having to take additional tissue wasted on a central test. That said, in the clinical side of things, I think we're really at a crossroads in clinical practice as to where FDA will fall in terms of regulation of laboratory developed tests, whether or not they'll cede control or continue to cede control to local departments of health and CLIA and to help modernize CLIA, or will adopt a more centralized review process for FDA. And that's currently remains unknown, and actually will have, I think, pretty large ramifications on the practice of molecular pathology in particular at academic medical centers, like where I used to practice and where a lot of us used to practice. And so, I look forward to seeing how that turns out. I think, you can clearly see from my soliloquy that I feel strongly one way, but I think in the next couple of years we'll figure it out.
You're entitled to your monologue. Scott, where do you see the future taking us?
Yeah, I like that future that Nino described, by the way. I really hope that can occur. I actually think that one of the paths or drivers will end up being when whole transcript platforms are authorized, and that will drive the requirement for this continuous learning, which is what we really need, and I think leverages what Nino says, because boy, I just couldn't agree more. It would be great if we could not have to send samples to a central lab, create concerns and inconveniencing patients, let alone the physicians, in having to do this. So, but I think that the whole transcriptomic sequencing is going to also be a driver where there's going to have to be a different approach there, because it's a little easier when you have variants to sort of take this, the current approach to regulation. But thanks for being on the panel. It's been great. Thank you.
Abdel, last words on the future of companion diagnostics and genomics.
I think it will increase by any mean. However, it's a long way, and there's a lot of work need to be done from science, implementation, regulatory, especially regulatory. I will not repeat what Anthony and Scott said and David, but one aspect that we did not touch on, sometimes probably part of the implementation and the attraction to big organization is the very low reimbursement, if any. Because, for example, while big LabCorp or Quest can charge $5 per blood glucose, they can make more profit than to do genomics, because the rate of failure and this relate to quality control and declare. Rate of failure in blood glucose runs, it can be 1 in 10,000 run. Lab probe may fail, but rate of failure in next generation sequence can be as big as 25%. So in order to have a sample reported, there is a lot of work and a lot of highly skilled people to do work. So reimbursement and the quality control are very important aspects as well.
Great. That'll be the substance of the next time we all get together. So sorry that we couldn't get to it, because as you point out, it's quite important. But I just want to echo the gratitude that I have for having you guys join and share your experience and insight and expertise. David, Abdel, Scott, Nino, thank you so much for a lively discussion. Reminder to all of our audience members, we'll follow up with an email with a recording. And if you have any questions or would like to reach out to myself regarding Genomenon and our offerings, or David and what Illumina is doing with respect to CGP, please don't hesitate. Thank you, panelists. Thank you, audience. Until next time.
Bye-bye.